Thank you, Ami, seriously, for that introduction and very caring adjectives and everything that you've said. And we'll see some of those photos again in my personal acknowledgements, strikingly. And of course, also thank you to the people who are attending in person and the people who are on Zoom. I really appreciate all of you coming to listen to my talk today. Today, I'll be talking about transmission of the human microbiome from infants to infections. The microbiome is a collection of microorganisms that live on and inside the human body. The greatest density and diversity of microbes are in the human gut, and we call this the gut microbiome. Over 10,000 species of bacteria, fungi, viruses, and archaea can live in a single person's gut. These microbes total roughly the same number of human cells in the human body, but in total, they only weigh around 200 grams. They're on average much smaller than human cells. Where the microbiome really stands out, though, is the gene content. Humans only have about 22,000 genes in the genome that contain instructions for making every cell type, but collectively, the microbiome has more than 150 times this number of genes. There's a wealth of unexplored functional diversity living right inside the gut. For a closer look at the gut microbiome, I love this microscopy image out of the Sonnenberg lab here at Stanford. This is a section of mouse intestines where the host intestinal cells are labeled in blue, the mucus layer excreted by the intestinal cells is labeled in green, debris from food is labeled in yellow, and the bacterial cells are labeled in red. What I hope you can appreciate is how abundant and pervasive the gut bacteria are. They live on and inside the mucus layer, right next to the host intestinal cells. All these bacterial cells living within the gut aren't just passive bystanders. Collectively, the gut microbiome has massive impacts on human health. These impacts include regulating the development of the human immune system, extracting certain nutrients from food that the body can't process on its own, protecting the human body from infection, affecting how the body responds to certain cancer therapies that can reactivate the immune system to target tumors, and affecting the brain and the nervous system, including producing up to 95% of the body's hormone serotonin. The gut microbiome is also dynamic and changes over time. Starting from the first days of a baby's life, the microbiome develops an increasingly complex community. Most of the first microbes colonizing the gut in an infant are the result of direct transmission from the mother. This direct transmission is especially important for colonization with the bacteria Bifidobacterium infantis, which has the ability to digest nutrients in human milk that the body can't break down on its own. Whereas close contact between a mother and infant is responsible for healthy microbiome development, other exposures during infancy and childhood can have a negative effect. For example, C-section birth and formula feeding can hamper the development of a healthy microbiome, but these changes aren't permanent. Antibiotic treatment and malnutrition early in life can also have a detrimental effect. Once a human reaches adulthood, most of the microbiome maturation is complete and the community is well established. It's understood that barring major modifications like a fecal transplant or extensive antibiotic use, the adult microbiome doesn't change much. Even drastic changes in diet, which can change the communities in the short term, haven't been shown to have a long-term effect. The developing microbiome is best understood as a continuum from infants to adults. As complexity increases with age, acquisition of new microbes falls. Fundamental research from Frederick Backhead and Marina Sour and others showed with precise genomic sequencing that direct transmission from the mother is responsible for the bacteria colonizing the infant gut. Because these microbes pass from mother to infant, we call this vertical transmission. Other research has shown that infants in the neonatal ICU can develop a microbiome similar to the hospital environment and filled with potentially pathogenic organisms. Both of these pieces of evidence point to the fact that the infant microbiome is quite malleable and often picks up new strains from either the mother or the environment. Moving up in age through adolescence and into adulthood, the gut microbiome community becomes established and it doesn't change much. However, there is some early research suggesting that transmission of gut microbes can occur between adults as well. Early studies suggested that humans could share bacteria with their intimate partners as well as their pets. As these subjects are unrelated, we call this horizontal transmission. More recent work looked at isolated villages on the Fiji Islands and showed that people living in the same household are more likely to share gut microbes than people living outside the house. The researchers could even predict spousal relationships on the basis of gut microbiome data alone. However, this doesn't necessarily prove transmission happened between adults because the authors only collected a single sample from each individual, and they couldn't really show that the same strain was present in an individual prior to a transmission event. So this leaves us with a big question mark about the transmission of gut microbes in adults. Researchers, myself included, think it probably happens, but there really isn't enough evidence to say one way or the other. Transmission between adults could be good for health if it spreads bacteria that are good for digestion, for example, or it could be a bad thing if it spreads antibiotic-resistant bacteria that can cause problems. Throughout my PhD research in the Bot Lab, I attempted to shed light on two parallel aspects of the microbiome transmission problem. To do this, I studied the two ends of the infant to adult spectrum. First, I studied if anything other than bacteria could be transmitted from mothers to infants. We found that a phage, which is a virus that infects bacteria, was commonly shared between mothers and her infant. Then I studied adults who were hospitalized for hematopoietic stem cell transplantation. These patients are treated with chemotherapy, radiation, and are often treated with antibiotics, and therefore have very perturbed gut microbiomes. We searched for transmission between patients who were hospitalized and were roommates as they recovered from transplant, and we found a few interesting cases of this happening. Before we get into the results of this research, I need to first explain how we study the gut microbiome. It all starts with a stool sample collected from a subject of interest and usually stored in the freezer. This sample contains all the bacteria, fungi, phages, and other organisms living in the gut. Inside each of these microbes is a DNA genome, typically circular, and it's made up of the DNA letters A, T, C, and G. So we extract that DNA into solution, shear it up into small fragments, and read out the DNA letters on a sequencing machine. From this experiment, we typically get several million sequencing reads, each around 150 base pairs in length. These sequencing reads describe the microbial genomes that were in the sample originally. And bacterial genomes are generally millions of base pairs long, so these small 150 base pair fragments are only a small section of that entire genome. From these millions of sequencing reads, we want to get answers about the microbial community that was in the original sample. The first thing we can do is compare these 150 base pair sequences against a database of all known microbes. From this, we can answer the question, who's there and in what proportion? We can also take the 150 base pair sequencing reads, find overlaps between them, and use this information to reconstruct larger microbial genomes. For example, we can take all of the dark blue reads here and reconstruct the 3 million base pair genome of E. coli. We can also look at microbial genes, either by assembling them from sequence or comparing against databases. And here we can identify 3 different genes responsible for antibiotic resistance. Finally, we can attempt to predict functional pathways that are present in the microbes that we're studying, such as the ability to break down certain nutrients. This type of analysis moves the question from who's there to what are they doing? Now that we have the capabilities to investigate the different organisms in the gut microbiome via metagenomic sequencing, we can move to the first research highlight of my talk. This work was titled Acquisition, Transmission, and Strain Diversity of Human Gut Colonizing Crast-like Phages and was published in Nature Communications last year. We already know that mother-to-infant transmission of bacteria is common and well studied. Frederick Bachhead and Marijn Jasor studied mother-to-infant pairs, collecting stool samples from the mother 3 months before, on the day of birth, and 3 months after birth. Samples from infants were collected at the time of birth and at regular intervals afterwards. The stool samples were analyzed with metagenomic sequencing, and the authors studied how different bacterial strains could be passed from mothers to infants. However, the authors only studied the bacterial fraction of the microbiome. Our lab was interested in the non-bacterial fraction of the microbiome, and transmission of things like phages was completely overlooked in this research. Luckily for me, all the data from these papers was made publicly available, and this enabled a computational researcher like myself to easily download the data and ask questions without having to process any baby poop in the lab. So around 2018, when I joined the Bot Lab, we were getting excited about a phage, again, which is a virus that infects bacteria, called crastphage. And crastphage is pictured here on the left. It has a head which contains the DNA genome and a short tail, which attaches the proteins on the bacteria and allows for infection. Interestingly, crastphage was completely undiscovered until 2014, when researchers pooled many different microbiome samples and looked in the fraction of the sequencing reads that could not be classified to any known organism. This process was called cross-assembly, and that's where crastphage got its rather unfortunate name and capitalization scheme. Crastphage could also not be isolated and cultured in a laboratory setting until recently, when researchers found that it infected bacteria in the genus Bacteroides. What was really surprising to us was that crastphage is very prevalent in human guts. Some estimates place it into about 70% of healthy individuals having a crastphage in their gut. It can also be very abundant, sometimes making up 10 to 20% of the sequencing reads from a sample. So it's really surprising that this phage was completely undiscovered until 2014. When researchers have looked for correlations between crastphage presence and human health, most of the effects have turned up negative. And there's been some associations between having more abundant viruses and inflammation in the gut, but this really hasn't been traced back to crastphage specifically. So it's possible that crastphage is simply replicating on the bacterial fraction, influencing how these different bacteria grow without having any real consequences for the host. And more recent research has shown that the singular Cras phage that I'll talk about is part of a much larger group of Cras-like phages. So throughout this talk, to avoid confusion, Cras phage is the one phage, and Cras-like phages is the much larger group. So with our interest in Cras phage in the developing microbiome, we started to wonder if Cras phage was vertically transmitted from mothers to infants, much like bacteria was. Nobody had asked this question before, and I have to credit Fiona Tamburini in the lab with coming up with the idea to look at these data sets in the first place. So we searched for Cras phage in the mother and infant samples, first from the Yassur study, and we found that almost 20% of mothers had the phage present in their gut at the time of birth. In the infant samples from these studies, we found Cras phage present starting at 1 month and increasing in prevalence up to 3 months, which is the final time point from this study. However, the fraction of infants positive for the phage didn't reach the level of mothers by the end of the sampling period. So it may take longer than 3 months for all infants to be colonized with Cras phage. We saw something similar in the data from the Bhatt study. 25% of mothers had the phage present at birth, and no infants had the phage present at birth or at the first sample time point. Presence increased at the 4 and 12 months time points, but still didn't reach the prevalence in mothers by the end of the sampling time period. So it's important to note that these studies were conducted in different populations and years apart, so you can't directly compare the proportions between them, but the major conclusions and trends remain the same across the two studies. So we showed that infants can acquire Cras phage starting at 1 month of age, but this doesn't necessarily prove that the mother was the source for that phage. We had to compare the genome sequences of the Cras phages from the mothers and infants to answer that question. So we used the process of metagenomic assembly that I described earlier to generate these larger Cras phage genomes. We found overlaps in the 150 base pair sequencing reads, and then we assembled the larger genome, which is roughly 100,000 base pairs in length. We then looked at the assembled genomes from mothers and infants and compared them against each other. By aligning the 100,000 base pair sequences and looking for places where there were mismatches, we can calculate a metric called average nucleotide identity, or ANI. And we found that when mothers and infants shared a similar genome, it was really, really close, approaching 100%. So we set the identity threshold for finding potential transmission at 99.99% ANI, or allowing for 10 mismatches across the 100,000 base pair genome, which could be due to differences in the genomes or some technical artifacts in the sequencing data. If we saw genomes that were less similar than that threshold, like the bottom example here, we reasoned that these strains were different and mother-to-infant transmission was not occurring, or perhaps these samples could have been from different families. So onto the heat maps, which are, as Ami introduced, is probably my favorite way to describe data. The alignment method that I used only compares 2 genomes at the same time. So we compared all possible pairs of genomes and then used this heat map method to visualize this data. And a heat map is basically just a color-coded matrix where each square represents the alignment % identity from comparing 2 samples. I've graded out the squares along the diagonal because by default, comparing a genome against itself has 100% identity, and that's not informative. And the matrix is also symmetric, meaning you can look at either the top or the bottom triangle. So for example, looking at the value for the infant and mother samples from family 1, we would look at this square here and see that it's bright yellow. From the color key, this represents 100% identity. We would say that the mother and infant had the same Cras phage strain. Comparing the infant from family 1 and the mother from family 2, we would look at this square, and green value is somewhere around 97% identity, and we would say that these phages are different, as we would expect from different families. So here's the big heat map comparing the results from all pairs of samples. I put colors along the sides of the heat map to indicate what family the samples came from, and clustered the rows and columns so that the blocks of similar color and similar genomes come out together. We had 10 families where at least 1 mother and 1 infant sample assembled a complete Cras phage genome we could use for comparisons. And in 8 out of 10 of these cases, the mother and infant samples clustered together. In the 4 families indicated with arrows along the side, we found 100% identical Cras phage genomes between the mothers and infants, including samples in mothers before the infant was born, really strongly suggesting that transmission was occurring between the mother and infant. So it was already known that infants born via C-section have different microbiomes in the first year of life. When we looked for presence of Cras-like phages, so the larger Cras-like phage group, when we looked in infants who were born vaginally, we found that almost 50% of them acquired a Cras-like phage by the end of the sampling time point. In the same time period, only 10% of C-section born infants had acquired a Cras-like phage by the end of the time point. We're definitely dealing with imbalanced classes between these two sets, but the difference was really significant. This result begs the question, Is the active vaginal birth directly responsible for transmitting a CRAS-like phage from mother to infant? Because the phages can take months to appear in the infant microbiome, I would say that that's not actually the case. It's more likely that the vaginal birth seeds the bacteria necessary for CRAS-like phage colonization later, and it could be certain things like Bacteroides species. So the metagenomic assembly approach I took earlier was really good for comparing CRAS-like phage genomes in different samples, but it ignores a key feature of human microbiomes. If you have a pure culture of E. coli, indicated here on the left, all the cells in that culture are going to be identical, and any representative you pull from the sample and sequence is going to have the same genome. However, in the human microbiome, within each species, there can be several different strain populations. Each strain might have slightly different functional capabilities, like the ability to grow better on certain nutrients. The strain populations within a species might be anywhere from 95 to 99.9% identical. So when you sequence a microbiome sample, you get just an average across all of these different strain populations, and it makes the data much more confusing. So strain diversity had been thoroughly investigated in the bacterial fraction of the microbiome before, but no one had looked at phages. So we were interested in studying strain diversity in the CRAS phage population and understanding if different things happened with strain diversity as the phages were transmitted from mothers to infant. So we had to do some method development to work specifically with phages, which I'd be happy to talk about later. I looked at the different strain populations that were present in the mother samples, and I found that strain diversity was typically less than what had been observed in bacteria, but there were still a few different strains, indicated here with the phages labeled A, B, C, and D. In cases when we thought direct transmission was responsible for mother to infant colonization, we found a subset of the strain diversity in the newly colonized infant microbiome, indicated by the presence of only the A and B strains here. And this effect makes sense if you think of the mechanics of CRAS phage transmission. Only a limited number of physical phages can be transmitted from mother to infant, and this small number can't represent all of the diversity originally present in the sample. So in population genetics terms, we call this a founder effect. We quantified the strength of this effect by measuring the fraction of positions in a CRAS phage genome that had multiple alleles present, and shown here on a box plot in a log scale. We found that CRAS phage populations in infants had roughly 1/10 the strain diversity that was present in mothers. So that for the overall conclusions from this work, we found that CRAS phages can be acquired starting at 1 month of age. And there was definitely hints in the data that it could be much earlier, perhaps starting as old as 1 week, but we need much better sequencing data and deeper sequencing to really nail that down. Next, we find evidence for identical CRAS phage genomes in mothers and infants, strongly suggesting that direct vertical transmission is responsible. And this raises the question that CRAS phage could be a marker for the maternal lineage, much like mitochondrial DNA is over many generations. So nobody had characterized strain diversity in the microbiome before, and we were able to show that CRAS phage strain diversity was less than what we found in bacterial cells, but still present, and we were able to show that strain diversity was reduced upon transmission from mother to infant. Future directions of this work, I'd really like to understand if other family members are involved in this CRAS phage transmission. No one has looked at father microbiomes or siblings or even other playmates of the infant, for example. I think it'd be really interesting to see if these strains are shared beyond the family. It's possible that we could use this direct transmission system to find hosts for new phages. And that's because if the phage engrafts in the infant microbiome, the bacterial host must also be present. And I use this system to look for the host of CRAS phage, and really the stats didn't allow me to say anything conclusively, so that remains to be seen in future data. We can use the infant gut to study strain colonization in general, and we can start to observe things like priority effects, which describe which bacteria must be present in the microbiome before a new bacteria can colonize. Finally, I'd like to look if other phages besides CRAS phage are transmitted from mothers to infants. I certainly looked at the phages that were known to be present in the microbiome and didn't find anything. But again, like the initial discovery of CRAS phage, there's definitely things that we're missing in this data. And Navami, who's a talented undergrad in the lab, is going to be researching this question now. So for high-level lessons from this work that really affected the future of my PhD research, I think it's important to look in the places where others had missed. And much like the initial discovery of CRAS phage, we looked for mother to infant transmission just because people had ignored that up until now. I also found that public data is a great resource to ask a question, and ask a question quickly for a computational researcher like myself. I started to think that microbiome samples hold an archaeological history of events up until that point. Every bacteria that's present had to come from somewhere, and perhaps by comparing these samples and unearthing what's happened, we can understand how colonization happens in new infants. And finally, I started to realize that strain level analysis was the future of microbiome science. We need to move beyond the species level and really understand the precise genomic sequences that are present in the microbiomes of the patients we're studying. So coming back to our model of microbiome complexity and the association with age, my research found that mother to infant transmission occurs with phages in the microbiome in addition to what was already known about bacteria. Thanks to the unique samples collected by the authors and the strain-specific methods I used here, we were able to deeply characterize this phenomenon. After this work, I was still interested in the adult side of the spectrum. It was clear that transmission between adults was rare. Otherwise, the earlier studies I talked about would have found something much more interesting. Therefore, I decided to study the problem in the most extreme case possible. Patients who were hospitalized for long periods of time with frequent antibiotic exposure that would kill off natural colonizers and make acquisition of more strains potentially more likely when the treatment ceased. So this next work is titled Rare Transmission of Commensal and Pathogenic Bacteria in the Gut Microbiome of Hospitalized Adults. And this work is currently under review at Nature Communications. In the Bhatt lab, our interests extend beyond genetics and into the clinic. We study patients undergoing hematopoietic stem cell transplantation, or HCT, which is a potentially curative therapy for heme malignancies like leukemia and lymphoma. The HCT process often involves transplantation of donor bone marrow, and patients are in the hospital for periods of weeks to months. There are many complications in the process, including infections, which are responsible for 22% of deaths following HCT. The high burden of infection necessitates frequent antibiotic treatment, both prophylactically and for treatment in response to these infections. The antibiotic treatment can really wreak havoc on a patient's microbiome. So we believe that the microbiome matters for HCT patients, and that by studying it, we could potentially improve outcomes for these patients in the future. As for why the microbiome matters, consider this clinical example. We collected 5 stool samples from this patient while they were in the hospital over a period of 2 months. This patient underwent transplant between the first and second sample. The microbiome composition in each sample is represented here by a stacked bar histogram, where the color of the bar represents the species that it was present, and the relative height of the bar represents the relative abundance of that bacteria. So I hope you can understand that this patient, when they came into the hospital, they had a healthy, diverse microbiome with lots of different species here and lots of species in the genus Bacteroides. However, with the third and fourth sample, this patient experienced intestinal domination with a pathogenic E. coli strain, indicated here in light purple, and it's made up to 80% of the sequencing reads coming out of this patient's gut. This patient then experienced an E. coli bloodstream infection, and thanks to work by Fiona Tamburini, who sequenced the genome of the E. coli bloodstream infection and the E. coli present in the gut, she found that they were totally identical, and that indicated that this E. coli probably translocated across the intestinal barrier and caused the infection in this patient. Thankfully, after antibiotic treatment, this patient recovered both from their infection and their gut microbiome recovered diversity as well. So after reviewing this example, I was left with the question, where did this E. coli come from? If we knew the source of this pathogen, we could work to eliminate it and provide better protections to our patients in the future. There are 3 possibilities for the source of this pathogen. It could be present in the gut of this patient when they came into the hospital, and if that was the case, there's really not much we could do. It could have come from the hospital environment because pathogenic and antibiotic-resistant strains are known to colonize surfaces and sinks in the hospital. And finally, perhaps most interestingly, this strain could have come via horizontal transmission from another patient in the hospital. And as I thought about this problem more, I realized that HCT patients are prime candidates to study horizontal transmission of gut bacteria. And that's due to all the features that I've been talking about, like the long stays in the hospital, periods of weeks to months, the conditioning process for transplant, which involves chemotherapy and radiation, the immunocompromisation during the transplant, which might alter the way the immune system responds to new bacterial colonizers, the frequent antibiotic use that kills off all the natural colonizers of the gut, and finally, that patients often had roommates in the hospital while they were recovering from transplant. So I believe that we can characterize horizontal transmission by studying HCT patients, and I designed a study to examine the microbiome of patients who were roommates as they were recovering from transplant. We began by examining the electronic health records of all patients who were in the hospital over a period of 3 years. Shown here on the left is a blueprint of the HCT ward at Stanford. There's 18 rooms, and there are 4 double occupancy rooms underlined. And in these rooms, patients have roommates. So because we had the electronic health records, I knew which patient was in which room at what time, and I could build a network of all patients who were roommates over the 3-year time period. Shown here on the right is the network, where every patient is a node, and I've drawn an edge between patients if they're roommates for at least 24 hours. And the width of the edge represents the total time of the roommate overlap between those patients. What I hope you can appreciate was that 77% of patients had a roommate at least once as they recovered from transplant. And 77% of the patients who had roommates fell into the largest component on the top of the graph here. This is potentially a network along which infections could be spreading over the period of 3 years. So in the Bot Lab, we've been collecting weekly stool samples from HCT patients going back to 2015. And to date, our collection has over 2,000 samples from 900 patients. And roughly 300 of these samples have been sequenced as part of other projects in the lab. So I picked 96 new samples to maximize coverage of roommate overlaps on this network. With these samples, I did a high molecular weight DNA extraction and performed an advanced metagenomic technique called linked-read metagenomic sequencing. This technique gives us roughly 10 times better genomes than what we could do in previous methods, 10 times better assembled genomes. From the combined collection of 400 samples, I generated roughly 1,500 unique species-level metagenome assembled genomes, or MAGs. And this collection of MAGs represents all the bacterial diversity that was present in our sample collection. When we tried to directly compare the assembled genomes, we realized that strain diversity within the microbiome was a major confounding factor. And the methods I developed to study strain diversity in phages really weren't up to the task of the more complex bacterial genomes. Luckily, Matt Ulm, who is a PhD student in Jill Banfield's lab at Berkeley and is now doing a postdoc at Stanford, developed a tool called InStrain that was up to the task of comparing these more complex bacterial genomes. To illustrate how InStrain works, imagine you have the same species present in 2 samples, and at a given place in the genome, 70% of the sequencing reads from sample 1 have an A and 30% have a T, and the sequencing reads in sample 2 have the reverse. If we perform the way that we typically compare bacterial genomes, which we call consensus SNP calling, we would say that the consensus allele, the 70% A and T, differ across these 2 samples. And we would say, yes, there is a SNP between these 2 samples. The innovation of InStrain was that he developed this method called population SNP calling. And here we say that these 2 samples share at least one allele. Therefore, they are indistinguishable, and we shouldn't call a SNP here. We found that the InStrain population average nucleotide identity, or POP-ANI, method works really well with the high levels of strain diversity present in the bacterial cells of a human microbiome. And based on comparisons where we compared the same strain over time in an individual, or positive control comparisons where we expected transmission to occur, like mothers to infants, we set a threshold of 99.999% POP-ANI, so nearly 100%. And we set that threshold for evidence that the same strain was present in multiple samples. So in short, we mapped sequencing reads from 400 samples against our 1,500 unique species-level genomes, and we compared strain populations between all pairs of patients. For a sneak peek at the results, we found relatively few cases of transmission, and this was surprising to us because we thought HCT patients would be prime candidates for this horizontal transmission. We found likely transmission of vancomycin-resistant Enterococcus faecium bacteria between patients who were roommates, and this pathogen transmission met our expectations. We also found nearly identical strains in patients who were not roommates, and this may represent acquisition of common hospital-acquired strains. We found likely transmission of commensal microbes as well, including Hungatella hathawayii and Akkermansia muciniphila. This was unexpected and perhaps more interesting. And finally, we found identical Lactobacillus rhamnosus strains in 8 patients over a period of 3 years. It was totally unexplained by any roommate sharing. So this was confusing, and I'll explain the results of this later. Starting with the transmission of pathogens, we studied the microbe Enterococcus faecium, which is a gram-positive coccus, and is a facultative anaerobe that can frequently develop resistance to the antibiotic vancomycin. And Enterococcus faecium is the most common pathogen to dominate the microbiomes of our patients, and it's the 3rd most common cause of infection in HCT patients as well. Here's one example where we observed transmission of Enterococcus faecium between patients who were roommates. These patients spent 14 days in the hospital together and 10 days in the same room. We collected 2 samples from the patient on the top on day 0 and 14. We collected 1 sample from the patient on the bottom on day 14. The microbiome composition is again indicated here with the stacked bar histogram. What I hope you can appreciate is the patient on the top had a microbiome completely dominated by Enterococcus faecium, shown here in blue, and making up to 90% of the bacterial community in this patient's gut. The patient on the bottom only had this bacteria at 0.4% relative abundance, but it was still definitely present. And so when we compared the strain populations across these 2 samples, we found 0 population SNPs between the 2 strains, indicating that we found 100% POP-ANI, there were no fixed differences between the strain populations, and we likely may have observed a transmission event between these two patients. So we don't have extensive time course sampling here. We can't say what this patient had in their gut prior to this single sample. I wish we had more, but my hypothesis for this is really transmission was responsible. For some commensal transmission examples, we studied the organism Hungatella hathawayii, which is a gram-positive rod and an obligate anaerobe, meaning it can't survive in oxygen. However, this bacteria can form spores to survive outside the host, which is what allows it to be transmitted between people. This bacteria is also known to induce regulatory T cells and suppress inflammation. So it's interesting that this gut microbe might be helping patients as they recover from transplant and often have gut-related inflammation problems. For our example of transmission of Hungatella hathawayii, these patients were in the hospital together for 34 days and spent a single day as roommates. We collected 2 samples from the patient on the top. This patient had Hungatella at around 5 or 10% in those 2 samples. And we collected 8 samples from the patient on the bottom who had Hungatella at around 50 or 20% in the final 2 samples. So again, when we compared the strain populations across these two patients, we found that they were totally identical across the entire genome, indicating that we think transmission may be occurring between these 2 patients. And what was interesting about the patient on the bottom was that they experienced a bloodstream infection around day 20, indicated with the X on the graph. This patient was then treated with antibiotics, and their sample on day 34 was completely dominated by Enterococcus faecium. So it's possible that the antibiotic use killed off all the healthy microbiome colonizers and then allowed this new strain to come in after the antibiotics ceased. Another interesting point was that this patient had a different Hungatella hathawayii strain present initially. So same species, different strain. It was around 96% identical. So it's clearly not the same strain persisting over time, but it's possible that the earlier presence of this species enabled the patient to be colonized again with the same species later in time. For the evidence for our transmission event here, we had roughly 7 million base pairs of the Hungatella genome covered, and we found 0 SNPs between the 2 genomes and 0 population SNPs. If we look at the more stringent consensus level, we only found 15 differences out of 7 million. So that's a very high consensus average nucleotide identity as well, and approached 100% there. If we compare our assembled genomes, assembled between the 2 patients, shown on the right is a dot plot where a strong line along the diagonal indicates that these genomes are identical across much of what we've been assembled. And there's not much structural diversity between the 2 genomes as well. But finally, we studied the organism Lactobacillus rhamnosus, which is a gram-positive chain-forming rod. It's a facultative anaerobe that produces lactic acid as a byproduct of carbohydrate utilization. And this bacteria is frequently used as a probiotic to help with digestion. When we looked at Lactobacillus rhamnosus in our patients, we found totally identical genomes between 8 patients over a period of 3 years. Shown on the left is a phylogenetic tree where all the identical genomes are highlighted. And on the right is a timeline of when all these patients were in the hospital and when we found the identical genomes. So interestingly, none of these patients were roommates and they weren't even in the hospital at the same period of time. So how did they all get the same bacterial genome? We dug a bit deeper, we found that all patients acquired this strain after transplant. Shown on the bottom is an example where this patient underwent transplant on day 2, and this Lactobacillus rhamnosus only came into the microbiome on day 16 and 20. And was then lost in future samples. And this mirrors what we know about probiotics, in that they often persist in the microbiome for a short period of time and then go away after you stop taking the supplement. So I think a commercial probiotic is responsible for this event. It's really interesting though, because patients are not allowed to take probiotics while they're in the hospital and recovering from transplant. They're also not allowed to eat high bacterial foods like active culture yogurt. So even though that is my hypothesis, there may be some patients who aren't quite following the rules in the hospital. For key takeaways from this work, we found that HCT patients frequently harbor pathogenic bacteria in their gut microbiome. This mirrors what was known from previous studies by other groups. We found that pathogens may be rarely shared between patients, and this includes Enterococcus faecium, but not E. coli. We found that commensal transmission was limited to specific conditions, including antibiotic exposure after infection and patients who are roommates with a shared bathroom. And then I'll pose the question that, is having a roommate as you recover from transplant necessarily a bad thing? It could spread infections, but we found that commensal transmission was almost more common. And if it spreads bacteria that help you recover microbiome diversity and suppress inflammation, it may actually be a good thing. For future directions from this work, I'd really like to perform environmental sampling in the hospital around the time of transmission. That would help understand if bacteria were found in reservoirs in the hospital or where the actual microbe was on a surface before it colonized the new patient's microbiome. I'd like to perform a prospective study involving day of admission sampling so we can understand what microbes are really present in the gut when these patients come into the hospital rather than just kind of guessing. We need to replicate these results in another cohort, perhaps at a different hospital, or maybe with pediatric HCT patients because they might be more likely to acquire new microbes. And finally, the HCT ward at Stanford has now moved to all single rooms in the new hospital, so it'll be interesting to repeat this experiment and see if we still observe any transmission. So coming back to the model, where microbiome complexity increases over time as acquisition of new microbes falls, my work has added that mother-to-infant transmission is common with phages in addition to what was already known about bacteria. I've also shown the first genomically characterized time course resolved example of microbiome transmission between adults. And we'll show that adults can transmit microbes between them, but it's a rare event and typically only occurs when the microbiome is perturbed by antibiotic use and patients are roommates in the hospital. So this work raises a high-level question. Why is transmission between adult microbiomes so much less common than what occurs between mothers and infants? I like to think of the two contexts like this. The infant gut is like a barren planet, untouched by life until this point. Many new microbes can colonize this planet, and colonization happens rapidly and frequently. There are still rules about what species can grow, driven by things like nutrient availability, but for the most part, the infant gut is a blank slate. As the infant grows, the gut microbiome develops into a mature and established ecosystem. An adult microbiome is like a mature forest ecosystem. Every organism fills a niche, and there's strict competition for nutrients and light. You also can't change the forest very easily. If you try to place a putting green in the middle of a rainforest, for example, it'll easily be overgrown. After extensive antibiotic treatment, the community will certainly be damaged, much like a wildfire ripping through a forest. And the forest may look damaged on the surface, but underneath the layer of ash, there may be seeds and microorganisms just waiting for a chance to grow. And this may be a new opportunity for organisms to colonize, but overall, the community isn't going to change much. And you wouldn't see redwoods growing after a wildfire on the East Coast, for example. So this idea that the adult gut is resilient, even after the extreme pressures we have observed in HCT patients, the chemotherapy, the radiation, the infections, the antibiotic treatment, this was one of the most striking things I observed during my PhD. The rules we discover about how humans acquire new microbes, they're incredibly important to understanding how we can modify the microbiome in the future, either with specific drugs or engineered microbes, and how we can use these tools to treat diseases that have previously been inaccessible to medicine. So with that, I would like to thank all of you for attending, the people on Zoom for attending as well. Shown here is a photo of our lab when I joined. I've been using this photo in my talk for a couple of years now, and then I realized that only like 2 or 3 people are still in the photo between the one that we took a few weeks ago. So it's really been a pleasure working with the lab and everyone in the lab that's assisted with these work. It's listed here. My thesis committee, RN, Ben, Hua, Denise, thank you for attending. I'd like to thank the people in the clinical microbiology labs that helped with this work, collaborators at Stanford Medicine, and outside collaborators as well. And with that, I will take any questions.
All right, so with that, questions, and we'll also monitor Zoom for questions. Summer, and do you mind repeating it?
Yeah.
Has anyone studied strain evolution over time within a person's microbiome? Like, will the strains change over time?
Yeah, people have, and especially in the context of like antibiotic or reconstitution of diversity after antibiotics, there's like rapid diversification that happens. And so people have studied, including in Ben's Good's group, have done some extensive work on this. And it's really interesting. I definitely suggest, you read the papers. I can't summarize them all here, but it happens, and it happens rapidly in some cases, and it's always sort of happening. And we can only observe a small fraction of it, I think. Becca?
Really great talk. I have a question about the crash stages in the vaginal birth versus C-section group. Did you find any differences in sexual reproduction?
Yeah, that was a good question. The question was, in the crash-phage vaginal birth, C-section birth, where there was a difference between, in crash-phage colonization, did we find differences in Bacteroides species? And was that a way to find the host? Definitely found differences in Bacteroides species. And if you do a principal component analysis of microbiomes of infants, you'll definitely see a difference between C-section and vaginal births during the first year of life. So there's really obvious differences. We did look for differences that would explain the host. And again, those were really underpowered with the number of C-sections births that we had. So I couldn't say anything there. We have a question from the chat from Gavin Sherlock, who asks, for cases where there appears to be demonstrated transmission, did you look at what isn't transmitted? And does it correspond to abundance in the donor or not? Gavin, that's a really good question. I think the idea being that if you already have a specific microbe present, it couldn't come into the microbiome, and it wouldn't likely take over a strain that was already there. And I would say that I didn't look at that specifically, but I never observed it happening. So I think that kind of answers the question.
Similar question about relative abundance. And this goes back to the founder effect that you spoke about, where you saw transmission in your scheme of phages A and B from mother to infant. Were those always phages A and B that were abundantly present in the mother, or were there other features about phages A and B that you saw?
Yeah, it was generally the dominant strain, and like the kind of the dominant allele at each position was really what got transmitted. It wasn't always the case, but that was the trend I observed. Sorry, the question being, did you observe the dominant strains being transmitted from mothers to infants? And the answer being most likely yes. Summer.
Did we have the sampling to see if horizontally transmitted stuff persisted over time?
Yeah, the question being, did we have the sample density to understand if microbes that were horizontally transmitted persisted over time? The answer being only like the Hungatella example that I showed, it was like 2 weeks or so. We could show that it persisted. That's a really great question that we would love to investigate. You know, in this prospective study, we could make patients come back for a sample a few months later and understand if it is, you know, only like a probiotic that goes away after the roommate period. That'd be an interesting question. Aaron?
So you mentioned based on the fact that like you have crassphage sort of not showing up until a few months after birth, detectable, like one hypothesis was that like the mother sort of seeds the host first and then the crassphage come along later. Do you have any thoughts on like why that happens? Like why they don't show up as quickly?
Yeah, that's a good question. Being why does crassphage not show up so quickly, and thinking that the mother seeds the bacteria for the phage to colonize later. I think it's not necessarily like Bacteroides aren't the first colonizers of the infant gut, but vaginal birth I think really sets the whole community up for those colonization events to happen. So it might not be that the direct hosts are transmitted, you know, during the first few days of life, and it might take a few days later for the host to actually persist. So I think it's more of kind of like vaginal birth sets up the community for that to happen later. We have a question in the chat from my cousin Lindsay Jacks. Curveball question in honor of COP26. I don't know what that is. Has there been any work on the high impact of heat stress on the composition of the microbiome? And if yes, any implications for human health? That is a great question and not an area that I have any expertise in. I imagine it would, given that, you know, stress hormones have been shown to impact the human microbiome, but I don't know about heat stress directly. Also affects diet, depending on, you know, if your community is heat stressed, your diet might not be as good as it could be. And that apparently CP26 is a climate change conference coming up on the 1st of November. Thank you, Lindsay. Yeah, man.
I know it's the same line later too, but
yeah.
Were you able to look at accessory genes at all in your transmission work, or is that just still too hard to reconstruct?
In the bacteria, do we look at accessory genes? Yeah, so I didn't look at genes at all. I saw that the previous transmission work had like looked at genes as individual units, and I kind of wanted to take a gene agnostic approach, where I just considered the entire genome and really just sequencing reads as evidence for this genome, regardless of what they map to. So in that case, I didn't even look, but it certainly would be interesting, and we have all the data. So.